Hi, welcome to my TED Talk. I hope you enjoyed the 2.3 seconds of mouth movement. It won't be happening again. Today we are going to talk about DNA replication and gene expression. DNA replication occurs when the body grows or heals itself. The process begins when the DNA helicase unzips the double helix by breaking the hydrogen bonds between the base pairs. New complementary DNA nucleotides then fit into place through complementary base pairing. These new DNA nucleotides are then joined together by the DNA polymerase. Because DNA polymerase can only go in a 3' to 5' direction, the anti-parallel nature of the DNA creates a lagging and leading strand. The lagging strand creates Okazaki fragments that are then joined together by the DNA ligase. When the process is finished, these new DNA strands are called semi-conservative because they have an old and new strand. Now that we've covered DNA replication, let's move on to gene expression. There are two steps to gene expression: transcription and translation. Once again, we will need the DNA helicase to partially unzip a section of the DNA. DNA polymerase then moves along the DNA strand, making a complementary mRNA. When the process of transcription is over, the mRNA is released, and the DNA strands bind together again. The mRNA is then processed to remove non-coding regions called introns, leaving behind coding regions called exons. After the editing process, the mRNA moves out of the nucleus and binds to a ribosome. tRNA then carries an amino acid to the ribosome and binds to the start codon on the mRNA. The 3 bases on the tRNA is called an anticodon, which corresponds to the codon on the mRNA. After the tRNA reads the mRNA, it delivers the corresponding amino acid. This process is repeated several times to create a polypeptide. Once it reads a stop codon, a release factor cleaves the polypeptide and it begins to fold into a protein. I hope you enjoyed the 1226 hand-drawn frames of DNA and the things you can do with it. Goodbye. I will see you all in therapy.